Luca, the last universal common ancestor, the originator of this and that and every living thing on Earth. What was it? In order to see where life began, we have to look closer. A lot closer. This is the Endosymbiotic Theory. You might have seen this growing on your roof or on trees. Lichen, a rough green mat that looks like a fungus, are everywhere. But lichen aren't a fungus. They're what we call a symbiont, a collection of several organisms working together for their mutual benefit. In the case of the lichen, it's a fungi with a cyanobacteria, or algae. 115 years ago, in the Soviet city of Kazan, a young scientist became fixed on lichen. Konstantin Mereshkovitsky wanted to know whether lichen were alone. Were there other symbionts? On the 35th page of his pinnacle book, he left a clue. Maybe all higher organisms came from a symbiotic relationship from simpler ones. Now, before we go on, I want to make sure that we have a proper understanding of the tree of life. You see, until the 1970s, the tree of life was split into two domains. Domain Eukarya and Domain Prokarya. Now, Charles Darwin had made it clear that we all shared a common ancestor, and for over 100 years after his theory of evolution, life was split between the eukaryotes, like animals and plants, and the prokaryotes, or bacteria. Now, eukaryote comes from the Greek words eu, meaning good, and karyote, meaning kernel, good kernel. The pro in prokaryotes means before kernel. Even then, it was well understood that the last universal common ancestor was a prokaryote. It was before the good kernel. The main difference between the two domains were organelles. Organelles. They were organelles. Eukaryotes have lysosomes, Golgi bodies, endoplasmic reticuli, and nuclei. Prokaryotes do not. Prokaryotes do not have organelles. So Constantine's footnote was earth-shattering. He proposed the chloroplast, the organelles of a eukaryote, looked like cyanobacteria, a prokaryote. His conclusion: the chloroplast must have been a prokaryote originally. But our friend Constantine, well, it turns out he wasn't that good of a friend. He was an anti-Semite, depressed, and eventually he committed suicide. So a lot of his work was forgotten. Enter Paul-Jules Portier. In 1918, just a few hundred miles away from Kazan, a French researcher independently came to the same conclusion as Constantine. In Paris, Portier was studying rickettsia, a microbe carried by ticks which causes typhus, when he noticed they looked a lot like mitochondria. Maybe mitochondria too were once bacteria, he wrote. Portier, like Constantine, was immediately shunned and forgotten, and for over 40 years, 4 decades, this work just was forgotten. No one was talking about it. Until one day, a young researcher at Boston University had a thought. Her name was Lynn Margulis, and she is the founder of this theory. Margulis realized that most of the organelles in eukaryotic cells looked like prokaryotes. Now, unlike Constantine and Portier, Margulis had a background in microbiology. By the age of 19, she had graduated from the University of Chicago, and at that point she was studying mitosis at Boston University. Now, one day while studying mitosis, the way in which cells divide, Margulis noticed something strange. It seemed like mitochondria inside eukaryotic cells were dividing exactly like bacteria, prokaryotic cells, through a process known as binary fission. And more importantly, mitochondria seemed to have their own genomes which looked a lot like those of bacteria. Finally, there was substantive proof beyond just a conjecture. Microbiology was showing something incredible. Bit by bit, more and more traits collided. It seemed as though mitochondria and bacteria were one in the same. They were all exhibiting the same behavior, even the same appearance. It was almost as if a cell had swallowed a bacteria and made it an organelle. And that's exactly what happened. Her first journal was rejected from publication 15 times. And when it did get published, most of her colleagues told her her career was over. Because Margulis didn't just think that mitochondria in the chloroplast came from bacteria. She thought that all eukaryotic organelles, all of the organelles our cells have, came from ancestral bacteria. She called this the Endosymbiotic Theory. And over the past 60 years, something incredible has happened. Nearly every single one of Margulis' hypotheses have held up. It turns out that eukaryotic cells, the cells we are made of, are just a ton of hijacked bacterial components. They're working for themselves, and we use them to work for us. But wait, the story gets better. Remember when I said the tree of life was split into 2 domains until the 1970s? It's okay if you don't, but something happened in 1977 which changed all of that. It was in that year that George Fox, working with Carl Woese, said, no, 3 domains sounds better. They were studying methanogens, bacteria capable of producing methane, except they weren't bacteria. By looking at their RNA, the pair realized that they were significantly different from anything that had ever been studied. These methanogens were, according to Woese, archaea, a 3rd domain of life, separate from eukarya and prokarya. Now, archaea look exactly like bacteria, but many of their processes are similar to those of eukaryotes. Like Margulis, Woese and Fox were mocked for years after their publication. But just 5 years ago, over 100 years after Constantine's initial discovery, all of that changed, and it all came together. In 2015, an expedition hundreds of meters below the surface of the Arctic Circle, near a deep sea sulfur vent, found very, very weird archaea. And they sequenced them, and they didn't look like any of the archaea that had been sequenced before. And they called these the Loki archaea. Now, remember that we need a cell capable of eating the bacteria. And Margulis thought that that was just a bigger cell, maybe another bacteria. But in the past 5 years, we now think that it was actually an archaea. More specifically, one of those Loki archaea that was found hundreds of meters below the surface of the Arctic Circle. And the Loki archaea would have eaten a bacteria to make our modern eukaryotic cells. Now, what does that mean? That means that every single cell, from our cat to our fish to our dog to our bird to humans, is the result of a million-year-old ingestion from an archaea eating a bacteria for dinner.